Okay, good morning, friends. Rather, good afternoon. Now, because of some technical snag, we lost around 5 to 6 minutes. Okay, let us move to the actual topic. Of course, yesterday we discussed about various principles of plant disease management and also classification of those principles of plant disease management. So, a brief, a review of what we have covered yesterday. So the classification. So various principles of plant disease management. The various measures are various methods to control this disease were classified preventive by Wedgel in 1923. He classified these measures into two primary categories. One is a preventive and the other is curative. Preventive means our aim is to see that the pathogen does not come in contact with the host, or our crop is protected from the onslaught of the pests or the pathogen, or our godowns are protected from the infestation either from the fungal, bacterial, viral, or by the infestation of insects. So first one is what we call preventive. The other is what we call curative. Preventive means before the disease appears, or all of our efforts are such that the pathogen does not come into our area or does not infect our crop. So this include, there are 6 to 7 preventive measures are there. Of course, the cultural, the mechanical, the physical, the regulatory, the genetic, and immunity. All these are involved in what we call preventive. Prevention, as you know, as I was telling you yesterday, prevention is always better than cure. So if you take all precautions or preventive measures, you may not be exposed to the pathogen. So you may not get infected with the pathogen and you may not develop any symptom, or you will not suffer from any disease. Of course, in spite of taking all precautions, of course, nothing is in our hands because we are exposed to the environment. See, the air we take, the water we drink, the food we take, and the clothes we wear, the area we live in, or all have so many pests or so many pathogens, because these pathogens, they are microscopic. You cannot see them. They may be a fungal spore or a fungal organism, or a bacterium will be there, or a virus will be there, or a mycoplasma could be there. So in spite of taking all our precautions, so the pathogen may make an entry, may come in contact with the host, thereby it makes an entry into the host, and thereby it causes disease. So this is preventive. So when it makes an entry, of course, there will be a tough fight between the host and the pathogen. Of course, the loser will be ultimately the host. The host develops certain symptoms and ultimately suffers from disease. Wherein disease is nothing but it affects the pathogen's entry, affects your, your growth, your reproductive ability, your, your physiological functions, your enzymatic functions, your hormonal functions, they get derailed. So ultimately, your growth gets affected. Your crop shows stunted, leaves show so many symptoms, and ultimately your produce or the product, the seed or the fruit that you expect, the yield you expect gets diminished. Okay, so this is preventive and the curative. Of course, curative primarily is of two types. One is chemical, the other is what we call biological. Chemical means use of chemicals. So once your crop develops diseases, means you have to take all measures that the pathogen does not cause a lot of damage to your crop. Thereby, you will be put to great loss. So to minimize the inoculum, to minimize the damage, you resort to what we call use of chemicals. So these chemicals, you spray them, you you spray them, or you treat your seed before sowing, or you add or you sanitize, your fumigate your soil, etc. So using chemicals in order to checking the population, in order to minimize the damage, in order to protect your crop, is what we call use of chemicals as a curative measure. The other is biological. So the curative can be chemical, and the other is, of course, environmental friendly, is what we call a biological method. Of course, these are all methods. Of course, coming to the principles, whatever we discussed yesterday, the principles were avoidance, exclusion, eradication, and protection, immunization, and therapy. So these two are inclusive. Whatever the curative and the preventive measures we told, they come under this. So they're all not mutually exclusive. Avoidance, I told you, avoiding, taking all measures so that your pathogen does not make or come in contact with your host. So all your measures, maybe starting your place, or plantation time, or variety use, etc., is avoidance. Avoiding the pathogen to come in contact with host. Then exclusion. This I told you, a legally bound, wherein quarantine. See that the pathogen does not come into an area, into a place, or into a country where it was not found earlier. You take so many quarantine measures, or inspection, or certification, and the treatment. This is what we call exclusion. And third one is what we call eradication. This eradication is nothing but agronomic practices, various agronomic practices, that is various tillage, or your mixed cropping, or your crop rotation, your flooding of this, your field, or removal of all your alternate hosts or weeds. All these are what we call eradication. Then of course protection. Protection is once so see that the pathogen's entry is regulated or checked. For that, what we do, we erect certain windbreaks, or you erect certain strips, sticky strips, or repellent strips, etc. Or you expose them to various radiations. So this is what we call protection. Then immunization. Immunization is nothing but going for breeding. That is use of resistant varieties. Use of resistant varieties, etc. is called what we call immunization. Of course, the last one is therapy. Therapy is nothing but our curative, wherein you use chemicals, or you use a physical, energy, or biological means to check or to minimize the damage to your crop. Now, with this background, we move to the measures, that is curative measures. Now, curative measures. So now, as we discuss about two curative measures. One is chemical, the other is biological. So first of all, chemical methods. Now, these chemical methods, say in this, I'll tell you certain technical terms that very commonly appear in your objective type of questions. See here, many chemical products are available. They check the proliferation, or checking the spread of the inoculum, or the pathogen, or the pest. So here, the chemical you spray, or chemical you put in and around your crop as a protectant, that chemical attracts the attention of the pests, and it reduces them, and ultimately it destroys, it kills them. Thereby, it protects your crop. So such chemicals that are used to check the spread of the disease, or to keep the spreading of the disease under control, is what we call chemicals, chemical agents. So, are otherwise known as pesticides. Now, these pesticides, the history of the pesticides is very recent, of course. The use of pesticides in agriculture started, agriculture started in 1950s. Of course, now the pesticides, use of pesticides, use of your inorganic nutrients, that is fertilizers, is at a high consumption rate. So now as for your objective type, which is the crop that uses, use of maximum chemical pesticides. You'll be surprised, 50% of the chemical pesticide farmers use on the crop to protect is your cotton. So 50% of the chemical pesticides are used to protect cotton crop. It is followed by 18% in rice, of course, 14% in vegetables, of course, in plantation crops, around 8%. Cereals and pulses account for 7%, and your sugar cane. In sugar cane, we go for spraying of in chemical pesticides to the extent of 2%, of course. Others account for 1%. So, but there are pros and cons regarding the use of these chemical pesticides. Of course, by using chemical pesticides, you can protect your crop. You can check the spread of the disease. You can arrest the spread of the disease at once. You get quick results. But the thing is, this is positive side. But on the negative side, you speak, it spoils your environment. It spoils your ecosystem. And whatever the chemicals you spray, residues, contaminate your food, contaminated your water, contaminate your livestock, your dairy products, and your pollinators get eliminated. Now you see hundreds and thousands of bee species are lost because of overspraying of pesticides. Or your crows, sparrows, of course, you say sparrows used to be in large numbers. Now sparrows are no more. They are on the red list. So your crows, your sparrows, your dragonflies, your honeybees, they are getting eliminated. So which are beneficial for the agriculture. So in one way, you are reducing, you are protecting your crop. But the other side is many animals, many insects that are beneficial for agriculture also getting eliminated. Your ecosystem is getting spoiled. Your water is getting contaminated. Your air is getting contaminated. Your livestock, your dairy products are getting spoiled, are getting exposed to ill effects. So this, not only this, even your wildlife is also not safe because of the overuse of these pesticides. Now, based on the target organism, based on the pathogen or the pest against which you are using this, chemical, pesticide, they are variously named. See, for example, the chemical that you use to check the fungus infection is known as a fungicide. The chemical that you use to check the insect population is known as insecticide. The chemicals that you use against herbs or the weeds is known as a herbicide. The pesticide that you use to control bacteria are known as bactericides. The pesticide that you use against nematodes is known as nematicides. The pesticide you use against mollusks is known as molluscicide. And the other very commonly asked one is avicides. Aves means birds. The pesticides that you use to check the crop soiling birds is known as avicides. And the pesticide that you use against rodents is known, rodents means rats and bandicoots. So they are known as rodenticides. So, okay. So fungicides, bactericides, nematicides, viricides, molluscicides, avicides, rodenticides. These are some of the pesticides that are various named based on the target organism. So now, in addition to this, we have, certain what we call botanicals or plant-derived pesticides. Example for them is pyrethroids. Pyrethroids we extract from, members of Asteraceae and members such as your chrysanthemum, tacitus, etc. Or from tobacco, you extract an extract known as nicotinoids. No, nicotinoids can also be used, and nimbin, or neem oil can also be used as an insecticide or a pesticide. Of course, we have From Strychnos noxvomica, you extract what is known as strychnine. So these are some of the botanicals. You are extracting the repellents from the, or the insecticides or the fungicides from the plants. Hence, they are called botanicals. Okay, now other, there are certain very important what we call terms, technical terms are there. For example, active ingredient. Active ingredient means the active component of the pesticide. See, for example, if you go to market, you get DDT, you get Novacron, you get what we call DDT, Novacron, or monocrotophos, dichlorovas. These are some of the pesticides. Now, in that you find what we call active principle. The entire thing is not, it doesn't contain concentrated active principle. Say, good example, I'll tell you, you are acquainted with, what we call capsules you take. Capsules or tablets you take. So that inside the capsule, that outer coating is there, inside you find only little amount of your active principle. So also in tablet also you find only little an active principle. So that is what we call active principle. And the other thing that helps for easy spraying, for easy dissolution is what that is the material. That use of that material into what we call into a duster, or into what we call into granular form, into a emulsifier form, into a liquid form. That is what we call a formulation. So formulation has active principle, and for the easy dispersal, for the easy spraying of that, you use non-active principle. So that non-active part is what we call formulation. So this formulation has active and inactive principle that helps in the easy transmission of the active principle onto the target organism. So, and we have what we call, chemical names. So chemical, for each, these pesticide has a chemical name. That chemical name is based on, IUPAC, that International Union of Pure and Applied Chemistry. Those people give that technical name or the chemical name for that active principle. Then, of course, you have what we call formulation, I told you, or trade name. See, trade name, the principle is same. Now, good trade name is, for example, nowadays, Modi and other people, they're popularizing what we call, asking the people to go for generic medicines. So in generic medicines, they have the active principle. But the trade names are different. But if you go to a doctor, it is mandatory. But it is not being enforced. Hence, still they give only trade names, not the active principle. See, good example, I told you, paracetamol, paracetamol antipyretic for fever use. And for of course, the common one is, if I say I am suffering from, high temperature, then you advise me to take crocin. Or the other form is what we call, Dolo. Dolo was very popular during COVID-19. So this crocin or Dolo, they are nothing but they contain paracetamol, which is the active principle. So now trade names are different, but the formulation, the chemical name is same. Of course. So now the mode of action, how this chemical, acts when you use it against a target organism. What is the mode of action? The mode of action will be like this. Generally on entering, when you spray that, it makes an entry. It could be contact, it could be a contact, or it could be a systemic. So many are systemic. They enter into the plant body, they get translocated along with the your xylem and phloem to entire plant body. Such are systemic. Some are contact, for example, dust or seed treaters, etc. They remain where you apply. So such ones are locals or contact. So they remain where you have sprayed. This can be used as a protectant. See, duster. Say, for example, in our households, generally, what you do if there are, ants are there, generally you put what we call gemaxin. It acts as a repellent. So wherever that U or Lakshmana Rekha is there, it's a chemical. So if you draw a line there, it acts as a repellent and insects move away. So here, what the chemical does, generally it interferes with the enzymes. So various chemical reactions that occur, biochemical reactions that occur in organism, they get altered. Enzymes get affected, and they also affect your neurotransmitters. The insects, on feeding, when you spray them, they become ill. They lie down. They lose their senses. They feel difficulty in breathing. They become unconscious. So neurotransmitters, they become blind. So they block your enzyme systems and the neurotransmitters, and also they create what we call hormonal imbalance. And they may kill the young ones, or they may check the what we call hatching of young ones from the eggs. Or the young ones are the grubs, or the caterpillars. They are the great sufferers. They don't have resistance against these chemical pesticides. Okay, now we in our syllabus we have fungicides, we have bactericides, we have nematicides, we have fungicides, we have bactericides, we have nematicides, we have insecticides. These are the 4 groups of insecticides that are there included in your syllabus, and we move fast. So now these fungicides, the fungicides, as the name tells you, the chemical that acts against the proliferation of a fungus, which checks the spread of the fungus, which interferes with the sporulation of the fungus. And this, when you spray fungus, it attacks or it interferes with the cell wall formation. The chitin, the fungal cell wall is made up of chitin. So chitin formulation, the chitin synthesis gets affected. The enzymes involved in the chitin formation get derailed when you spray a fungus. Or they check even the enzyme systems and the reproductive process. The cell cycle gets affected. The enzymes involved in cell cycle and the cell wall formation, all these get derailed. Thus, their proliferation or the cell cell division or the sporulation or the reproductive methods get affected. So hence the spread of the disease gets affected, and your crop ultimately your crop gets affected, because you are with the spraying of this, you are arresting the dosage or the population of the fungus. So now the fungicides are classified variously. Now generally the fungicides are primarily applied as seed dressers. Pyram is there, Capton is there. You before sowing, or sulfur dust is there. What do? You treat your seed with that chemical. That what we call seed treatment. So you treat the seed, or you mix your soil or your soil bed or your greenhouse soil with the fungicide, so that it gets sanitized. Or you, what we call, when symptoms appear, you spray this fungicide on the infected areas, especially on the foliage or on the inflorescence, or the area which shows symptoms. So it could be seed applicable, soil application, and also foliage application. Now coming to the forms, in which form? In various forms, generally the fungicides are applied. One is what we call in the form of dust, that is in the form of powder, or in the form of emulsifiers, that is oil-like emulsifier. You add water and it becomes so whatever the concentrate you get that liquid form, you add into what we call water and you thoroughly mix it. That what we call emulsifier. Or in some cases, the fungicides are used as fumigants. Fumigants means here they emit certain fumes which are poisonous for the insects, for the host, so or for the pests. So that is so in the form of dust or emulsifiers or fumigants you can use. Now, the classification of fungicides. See, fungicides are variously classified based on their action. Some are what we call broad spectrum fungicides. Some are called narrow spectrum fungicides. Broad spectrum fungicides means they are capable of, see, for example, if you are using a fungicide against a mildew, it can also check the powdery mildews, downy mildews, or to certain extent even rust and smuts, etc. So such chemicals, such fungicides, which are capable of acting against various fungal pathogens, are known as broad spectrum fungicides. Narrow spectrum fungicides means for each disease, for each pathogen, you employ or you make use of a given fungicide. That is what we call narrow spectrum fungicides. Now, based on the mobility, see, based on the mobility, generally there are some are called what local, some are called what we call systemic. Local means they remain wherever you spray, the fungicide remains there itself. Such are called what we call local. Local ones, for example, Captan is there, Tyram is there, and Mancozeb. These are called local. They remain wherever you spray. So generally, we spray on the areas, infected areas. You make use of what we call these locals. And against this, there are systemic. Systemic are those, if you spray on the leaf, it doesn't remain on the leaf. It makes an entry into the leaf system, or it enters into your mesophyll. From the mesophyll, through veins, it spreads to the entire leaf, foliage, and also along the entire even stem tip, to the branch tip, etc. So such fungicides that move along with the water or the what we call through xylem and phloem tissues is what we call systemic. They spread to the entire plant body. See, for example, if you suffer from any disease, generally, if you go to a doctor, he gives you an injection. If you have any, say, for example, an injury, there you apply certain local medicine in the form of a paste, in the form of an oil or so. This is local. But if you take an injection, intramuscular, that spreads along with your blood, etc. It spreads throughout your body. So such are called what we call systemic. So local or systemic. Systemic example are, for example, Benomyl is there, and carboxym is there, or carbendazim. These are some of the what we call systemic fungicides. Okay, then fungicides, what we call based on their chemistry, based on the chemical nature, the fungicides are classified. So they are primarily classified into two, what we call the inorganic fungicides, and the other is organic fungicides. Inorganic means they have what we call a copper-containing fungicide, a sulfur-containing fungicide, a carbonate-containing fungicide, or a phosphate-containing fungicide. So such ones are called what we call inorganic fungicides. For example, Borax mixture. This is very, very important. Borax mixture, this is the first fungicide. It was Milordet, a Frenchman, who first invented this borax mixture. The borax mixture is nothing but copper sulfate and calcium hydroxide mixture. That what we call a copper-containing fungicide. And of course, a borax mixture is there, and copper oxychloride is there, and sulfur dust is there, sulfur lime. These are sulfur-containing. Of course, carbamate, sodium bicarbonate is what we call example for carbamate. And phosphates, of course, monopotassium phosphate is example for a phosphate containing inorganic fungicide. Then coming to the organic fungicide. The organic fungicides are plenty. Their numbers are very plenty. Of course, they are also divided into various categories based on the chemistry, that is organic chemistry. Now, based on that, they are quinone containing, they are aromatic compounds, they are heterocyclic compounds, etc., etc. And a big list is there for you. For example, organic, inorganic, some are sulfur containing compound. Example for that is what we call thiram. And of course, ethylene containing organic compound is macogeb. So macogeb is there, and gineb is there. And gineb's trade name is, of course, dithenium 78 is there, maneb is there, and it is manjed D. It is what we call its trade name, and generally they are available in the form what we call in the liquid form. Of course, except this thiram, or sulfur dust, they are available in the form of duster or in the form of wet powder. Then aromatic. Aromatic means sweet smelling. They smell and they evaporate. Aromatic compounds. See, good example for that is pentachloronitrobenzene. This is a aromatic fungicide. And heterocyclic compounds, for example, Capton is there. And organic systemic fungicides, for example, Ridomil is there, Metoxil is there, Benomil is there, Carbendazim. These are examples for organic systemic fungicides. Then, of course, organophosphate fungicides are there. And generally, the phosphoric acid is there, pyrimidines are there, triazoles are there. These are all examples for phosphate-containing fungicides. They are used against, powdery mildews or leaf spots, rust, and smuts. And strobulins are other category of fungicide. Generally, they are used to sanitize the soil or even to treat the seed before or seed-borne, to check the seed-borne diseases, and also to sanitize the soil. Generally, these strobulins or kind of fungicides are generally used. These are all what we call fungicides. So primarily fungicides are of two kinds. One is inorganic and the other is organic. Inorganic, quinones are there, heterocyclic compounds are, benzene containing or organophosphates are there, organochlorides are there. Good number of are there. So then second category of pesticides is what we call bactericides. As the name indicates, the chemical or the pesticide that acts against bacteria is known as a bactericides. Generally, bactericides. So in example, generally in exam under an objective, you get it. Which of the following is a bactericide? So generally, the antibiotics. So the antibiotics, the plant antibiotics there. So generally, example for this is, for example, agrimycin, agriculture, used in agriculture. Streptomycin is there, tetracycline is there, cycloheximidine is there. These are some of the examples for, bactericides. So generally, you use bactericides if your, crop is infested with bacteria. For example, Xanthomonas axinopodis citrus canker, or your Xanthomonas oryzae, bacterial blast of rice. So against them, generally, you make use of what we call these bactericides. Or Erwinia carotovora is there. That also, in wilts also. Wilts, Xanthomonas malvacearum. Then generally wilt causing. There also you can make use of this, bactericides. Then, under objective, you will also get what we call nematicides. Nematodes or roundworms. So generally, they cause what your root knots, or even they attack your root system of your tobacco. So there you can make use of what we call the chemicals that act against nematodes. Good example for them are, they are either used as soil treatments or also as fumigates. So generally, methyl bromide is an example, and chloropicrin is another example for a nematicide. And the last one is what we call insecticides. See, as the name tells you, the chemicals that are used to check the diseases that are caused by the insects. The insect causing, they cause a lot of damage, and also they act as transmitters of various viral diseases. Nephrotics, viruses, we have seen, Chungro virus transmitter. So to check them, you make use of insecticides. Now, insecticides, a good number of insecticides are there. Here also, based on the chemistry, some are what we call organochlorides, organophosphates, and carbamates are there. Say, for example, organochlorides. Good example is DDT. So sometimes they'll ask you to give the full form of DDT, dichlorodiphenyl trichloroethane, organochlorides. Of course, the organophosphates. Generally, dichlorovas is there, chloropyrifos is there, monocrotophos. These are all phos. They contain phosphorus. And carbofuran is a carbamate. So carbofuran is there, and phorate is there, chloropyrifos is there, monocrotophos is there, carboxyl is there, endosulfan is there, dichlorovas is there. So these are some of the, and not only that, dimethoate is there, methyl parathion is there, trizophos is there, methyl demeton is there. These are some of the insecticides that act against insects. So insects, we have, aphids, thrips, we have, you have, grasshoppers, you have. They cause a lot of damage. So to check their population, generally you make use of insecticides. So this is about the chemicals that are used to check various pests. So we discussed about 4 different groups of pesticides. First was a fungicide, second was a bactericide, third was a pneumaticide, and the fourth one is what we call a insecticide. Okay, then, so this is about insecticides. Of course, curative, I told you, chemicals as a curative mechanism, and the other is biological. So use of biological organisms to check to keep the population or the spread of the disease under control, such using living organisms against living organisms is what we call biological control, otherwise known as biocontrol. In chemistry, of course, in chemical pesticides, generally you spray chemicals. They may act against a bacterium, a fungus, a mycoplasma, or an insect. But here, the other is what we call in integrated pest management. Now, scientists and the environmentalists are stressing on to keep your environment free. I told you by excessive use of chemicals, we are spoiling our environment. You are damaging your aquatic ecosystems. You are damaging your sea ecosystem. You are damaging your so you are damaging your environment. So in order to check these pitfalls or the negative effects of excessive use of chemicals, now the agricultural scientists are popularizing or educating the farmers and other people, agricultural scientists, to go for biological method of checking the spread of the disease. So here you use biological organism to check the population of disease causing organism. So this what we call this is an environmentally friendly method, and which checks the spread of the disease. Of course, under this biological control, pest control, biological pest control, generally we discuss about what we call here, you encourage the natural enemies of the disease-causing organisms. Disease-causing organism, you check the population of disease organism by using other organisms that are present in the vicinity, in the local ecosystem. They're natural enemies. So here, the natural enemies could be parasites, the natural enemies could be predators, the natural enemies could be pathogens, the natural enemies could be competitors. So 4 different types of biological organisms are used to check the disease spreaders or disease causers. They are predators, pathogens, parasites, and competitors. We will discuss them now. Now, this biological control, of course, this I already told you is an environmentally sound and cost-effective and effective means of reducing or mitigating or protecting your crops from the onslaught of various pests and pathogens. Now, this International Organization of Biocontrol, there is an organization known as International Organization of Biocontrol that defines this, use of living organisms or their products, use of living organisms or their products to prevent or reduce the loss or damage caused by harmful organisms. So you are checking the population of harmful organisms by using their natural enemies or the products of their natural enemies. This is what we call biocontrol, using of biological organisms or living organisms, checking the population or the disease spreaders of disease-causing pathogenic organisms is the good definition for what we call biocontrol. So now, this in biocontrol, generally, we make use of this term mostly in entomology, that is study of insects, and also in plant pathology. So now, the biocontrol. In the biocontrol, there are 3 important strategies. 3 important strategies involved in biocontrol are what is called importation, augmentation, and the conservation. So sometimes you will get them under a short note. Importation, augmentation, and what we call the conservation. These are the 3 strategies that are involved in biocontrol. So what do you mean by importation? The term itself tells you. It gives you a clue. Importation. So what you are doing? So here the thing that is involved here is, I already told you, you are using the local enemies, the enemies that are available in the vicinity to check the disease-causing organisms. Say, for example, to check, mosquito menace, doctors advise you, in the areas where there is a stagnant water, they ask us to grow what we call a type of fish that eats the larvae of the mosquitoes, Gambusia fish. This is a kind of what we call a biocontrol. Okay, so now importation. So now here importation is, see, we have so many, crops or varieties you import. So you'll be surprised, most of the crops that we call, we feel, say, for example, your mirchi. Mirchi doesn't belong to India. Your groundnut doesn't belong to India. See, your what we call sunflower doesn't belong to India. It came to India and established in India. Now it suffers from various diseases. So we don't have its natural enemies here. So what we do is you go to the country from where it is where it is growing naturally, where it is supposed to have originated. There you find its enemies. So here you import the enemy from the country from where this crop or from where this disease-causing organism has come in. That is what we call importation. So here you import that natural enemy of that disease-causing organism in less numbers, of course. Then what you do is generally you inoculate. Inoculate means you release into that area so that their population increases and they check the spread of the disease-causing organism. This is what we call importation. The second one is augmentation. What do you mean by augmentation? Augmentation is nothing but periodic release of natural enemies into the surrounding, into the ecosystem. This is of two types. One is what we call inoculative release and inundative release. Inoculative means releasing the natural enemies in limited numbers at regular intervals. You release the natural enemies in regular intervals in limited numbers. Whereas in inundative means at a time you release these enemies in large numbers. So this is like you were spraying your chemical pesticides. So you use maximum dose of chemical pesticides so that you check the spread of the disease. So here in inundative method, you release these natural enemies in large amounts. This is what we call a augmentation. This is what we call augmentation. So then the third one is what we call conservation. So conservation is unlike the first two, that is importation and augmentation. The conservation means here you encourage the local enemies. In the previous two ones, what we did, you imported the natural enemies from the countries from where that pathogen came from. So here, what you do is you conserve, you encourage the natural enemies, you maintain these natural enemies, you maintain them in what we call beetle blanks, or you create a crop, a greenhouse, wherein you encourage the natural enemies, or you grow the alternate hosts. When the crop is not available, the enemies also may die. So what you do generally, you create a greenhouse. You grow, you create a microclimate there, a mini crop there. There you see that the population or bio agents are being maintained there. So whenever your crop gets infested, then you take these enemies and you release in that area. This is what we call a conservation. Then of course, this biocontrol. Now this biocontrol agents. So generally the biocontrol agents, I told you, generally the biocontrol agents, they may be of various types, They are primarily of what we call parasites are there and predators are there, entomopathogens are there, entomopathogenic, bacteria are there, baculoviruses are there, and entomogenic nematodes are there, and plant pathogen antagonistics are there. These are all various kinds of biocontrol agents. So based on the target organism, they are variously named. See, for example, first one is predator. What is a predator? Predator is an organism that spots the disease-causing organism and eats them, physically lifts them, physically eats them. This is what we call a predator. See, for example, predators are, for example, dragonflies are there, or beetles are there, the spiders are there, mites are there, monkeys are there, toads are there, birds are there, birds like crows, sparrows. So if you closely observe a field, there you will find, all these flies, dragonfly. It picks the young ones of the aphids or caterpillars, eats them. So also beetles, they also, the spiders, the monkeys, of course, they take them. And even, of course, your birds, crows and sparrows, they spots them and picks them, physically lifts them. So they are called predators. So they directly feed on the disease-causing pests. Such ones are called predators. Second one is what we call parasitoids. So the parasitoids are, parasite, host and parasite is there, of course. Host is the crop plant, parasite is disease-causing pest. So here parasitoids are, on the parasite, you inoculate, you see that its enemy is seen growing in or on that disease-causing organism. Say, for example, good example for this, trichogamma, a fungus. Trichogamma is a fungus. It is known to infect various insects. It is seen growing on the surface of the insects, but also into the tissue. It makes an entry into the tissue. There, when it grows fully, generally the disease causing pathogen suffers from growth, and also it loses its appetite. It becomes very dull. It feels very difficulty in breathing, and ultimately it dies. So a pathogen, a fungal pathogen growing on an insect and causing impairment in the insect is what we call a parasitoid. On a parasite, again, a smaller parasite is growing. That is parasitoid. Example for this is a fungus. Then entomopathogens. Entomopathogens means there are pathogens that are seen growing inside the body of an insect. It could be what we call, it could be fungus. A good example for this is entomopathogens are there. And these entomopathogens could be a fungus, it could be a bacterium, it could be a virus. Say, for example, first of all, fungal entomopathogens. A good example for this is Numura relayea is there, Bavaria bassiana is there, Trichogama is there, Verticillium is there, Metarhizium. These are various species of fungi that are seen growing on the body of the various insects or inside in the gut of the insects, and thereby they derail its its various metabolic processes. Thereby, finally, it dies. And you will see, you'll be surprised. If you visit any crop plant, there you will see some insects are dying. They hang, because if you closely observe, you will see certain fungi spreading over its surface. So they are the parasitic fungi. Then of course bacteria are there. For example, certain Bacillus thuringiensis is there, and this Bacillus thuringiensis grows, or certain protozoans are there. They grow on other, generally in cotton and in corn and in potato, etc. There, this Bacillus thuringiensis grows in the Lepidopteran insects and cause diseases in them. Thereby, they kill them. So here, what you are doing, you are not doing. You are seeing, you are spraying, you are dusting that area with this bacteria containing, pest. So it makes an entry into the body of the insect, and ultimately it impairs with its various life activities, and finally sees that the insect dies. Then, of course, baculoviruses. There are certain viruses, polyhedral viruses are there, nucleopolyhedral viruses, and granuloviruses are there. They are known to infect certain bollworms and tobacco caterpillars. If you spray these formulations having these polyhedral viruses, they make an entry into this bollworm or tobacco caterpillar, and thereby they may cause diseases in them. Ultimately, their population gets, and their reproductive ability gets affected. Then, of course, nematodes. Entomopathogenic nematodes are there. For example, Xenorhabdus and Photorhabdus. These are the two, bacteria. These two bacteria, they make an entry into the body of the nematode, the young ones of the nematodes. And these nematodes, they feed. Whenever they feed or whenever they enter, making an entry into the insect, these bacteria, they ultimately make an entry into the lepidopterans or the insect, and finally kills them. So even certain protozoans are known to infect parasites on insects. Sporozoans are there, and Nidospora members are there. They are known to cause death in various insects. And finally, plant pathogens. There are plant pathogens. So plant pathogen antagonistics. So there are certain species. They see our crop plants suffer from so many diseases. There are certain plants, there are certain fungi which act against the fungal diseases. Such ones are called plant pathogen antagonistics. Example, trichoderma is there, and gliocladium is there, bacillus is there, streptomyces, pseudomonas, etc. are there. They cause so many diseases in crop plants. So if you use these formulations having these pseudomonas or xanthomonas or bacillus or streptomyces species or trichoderma or gliocladium species, if you spray these formulations having those either bacteria or the fungal members, they are known to arrest the spread of the various bacterial diseases and also various fungal diseases. Such ones are called what we call the plant pathogen antagonistics. Okay, now, of course, certain antagonists are there. For example, certain weeds, say, for example, water hyacinth, what we see. Water hyacinth or parthenium is there, opuntias, these are all exotic. They are not natives of India, but they are posing a great problem to our water bodies. Water hyacinth, and Hussain Sagar, etc. There you will see water hyacinth growing. Parthenium, Parthenium hysterophorus, a member of Asteraceae, you will see growing on the hedges along with the crop plants. Now opuntia, prickly pear, this is growing in the barren areas. They are posing a lot of problem. So to check their population, what we do is, now scientists identified their natural enemies from the areas, and they have imported various fungi and rust-causing fungus, say, Puccinia, for example, abrupta is known to infect water hyacinth and thereby develop so many symptoms. And so also Alternaria eichhornea is the another fungus known to infect the eichhornea. And of course, there is Cercospora rhodani is there, and this also known to check various fungal diseases. Now, finally, what we do is, what are the advantages of biological control and what are the disadvantages? Of course, advantages are outnumber disadvantages. But the thing is, advantages, generally here the action is very slow. So what you are doing, you are releasing their natural enemies, but it takes time to develop, to build up their population. So their activity will be very slow. You don't get immediate results. So it's a long and time consuming. And the other is cost effectiveness is not affordable. And generally, they don't cause any poisons. So generally, for example, in chemical control, we have seen they are spoiling our water bodies. They are spoiling our livestock. They are posing dangers to the human beings. So, but in biological control, you don't see such things. So they don't have any residues left. Their action will be a bit slow, and they don't spoil your ecosystem. And your dependence on pesticides gets reduced if you go for biological control. And of course, very safe to apply. And of course, generally, the disease resistance will be at minimum. Then coming to the disadvantages is you need advice of the experts. Needs experts guidance because we are dealing with living organisms. And the other is, it is a very costly effect, and it takes long time. And by the time you lose your crop. So it doesn't eradicate complete like your chemical pesticides, a slow process, time consuming, long-term effects will be there, and a low economic threshold is the other thing. And awareness is very much needed. And generally, the other is the agent, generally the bioagent generally you use may may be intended to only a particular species. But in chemicals, we have seen if it is a broad spectrum pesticide, it kills not only that particular, but also other things. That is there. These are the advantages and disadvantages of the biological control. So we discussed about chemical control and biological control. Chemical control, of course, we have seen organic, inorganic, wherein they destroy your cell wall synthesis or they derail the enzyme systems, etc. Thereby, they check the spread of the disease. They could be inorganic or organic, etc. We have seen. Of course, the only good thing is you get immediate results. That is there. But the other side is it is spoiling your ecosystem. Dangerous to your human beings, dangerous to your livestock, and even wild species gets affected. But at integrated pest management or recent researchers and agriculture scientists advise us to go for biocontrol, wherein you use other organisms or the enemies of the pathogens to check their proliferation. This is what we call bio, and this bio is of we have seen predators, we have seen, it could be a bird we have seen, it could be monkeys we have seen, dragonflies we have seen, or it could be your plants against the plants, fungus against the fungus, or bacteria against the bacteria we have seen. So this is about what we call biological control. So if you have doubts, you can raise doubts regarding today's class. And the thing is, tomorrow we shall have last class, and try to attend the last class tomorrow. So we'll meet tomorrow from 12 to 1. If we don't have any query, then I'll call it a day. I'll end my lecture here. Thank you. Thank you for your patient hearing. Thank you.